Thank you very much. Good afternoon, everyone. My name is Fernanda Machado. I'm a researcher at the University of Aveiro in Portugal. And first, I'd like to thank for the opportunity to be here. And today, I'm going to show you a little bit of the work I have been developing in my PhD about the colonic fermentation of these coffee melanoidins and the resulting cardioprotective metabolites. So, as we know, coffee beverage has several beneficial compounds, namely polysaccharides, melanoidins, trigonelline lipids, chlorogenic acids, caffeine, among others. And these compounds are responsible for the numerous health benefits associated to coffee: the neuroprotective, immunostimulatory, anti-inflammatory, anti-diabetic, hypocholesterolemic, and microbiota modulation. And in this work, I'm going to explore a little bit the relationship between cholesterol-lowering properties of coffee and the activity of the gut microbiota, and how is this related. So, as we know, cholesterol is an ambivalent molecule. On one hand, it modulates membrane structure and function. It acts as precursor of biosalts, vitamin D, and hormones. But the excess of cholesterol in our blood, it can lead to the development of cardiovascular diseases, which are the leading cause of worldwide mortality and disabilities. So currently, there are several strategies to decrease cholesterol. Namely, we have statins, which decrease cholesterol synthesis through enzyme inhibition in cholesterol synthesis pathway. We have resins, which decrease cholesterol absorption through a biosequestration mechanism. We have ezetimibe, which is an inhibitor of cholesterol transporter, decreasing cholesterol absorption. And we also have phytosterols, which decrease cholesterol absorption through a co-solubilization mechanism. So these strategies, they have several drawbacks. They can be costly, they present severe side effects, and they have reduced efficiency and selectivity, and their intake time is not the most appropriate. So there's a need to develop more effective ingredients for this purpose. So coffee could be a good matrix for this. It is already part of people's daily habits. It's possible to modify its chemical composition by altering the processing, the roasting, the grinding, the brewing, and so we can affect coffee properties. It is taken after major meals, which is when we have more cholesterol present in the intestinal lumen, so we can have a higher effect. And it's rich in bioactive compounds. So in a previous work, we have seen that galactomannan and arabinogalactans, which are coffee polysaccharides, they can affect cholesterol solubility. So if we look here in this graphic, we can see that coffee beverage samples, together with coffee extracts that are rich in these polysaccharides, they are able to decrease cholesterol solubility when compared to a negative control. So we also have the melanoidins, which are compounds formed during the roasting of coffee. They are formed through the transglycosylations, esterification, and Maillard reactions, which are involving these polysaccharides and also phenolic compounds and proteins. So these melanoidins, they could also have the potential to affect cholesterol, since in their structure it involves these polysaccharides. So let's look a little bit on the mechanisms behind this effect. So cholesterol is synthesized in the liver, and then it's used to produce primary biosalts, which are then used to solubilize the cholesterol coming from the diet and the endogenous cholesterol produced in the liver, and in the micelles, helping to absorb cholesterol. And we have seen that this coffee dietary fiber, they are able to interact through hydrophobic motifs with these biosalts, sequester them, and then less of these biosalts will be available to emulsify the cholesterol. So cholesterol will not be so efficiently absorbed, increasing its excretion. Also, these biosalts, they are usually recycled back to the liver through the enterohepatic circulation, and when these fibers sequester them, they prevent them from going back to the liver, and the liver has to recruit more cholesterol from the bloodstream to produce more biosalts. Another mechanism is through fiber fermentation. So these fibers from coffee, they can be fermented by our microbiota, and they produce these metabolites, the short-chain fatty acids, and acetate and propionates, they are involved in cholesterol homeostasis. Namely, the acetate, it serves as the building block for cholesterol synthesis, and propionate inhibits the rate-limiting enzymes in this pathway. And so the ratio of these two fatty acids is really important to regulate cholesterol homeostasis. Another mechanism involves the biosalt biotransformations. So the small percentage of these biosalts that escapes the enterohepatic circulation to the liver, they can go to the colon and be transformed by the bacteria to secondary biosalts. These secondary biosalts, they are more hydrophobic, so they can better emulsify the cholesterol. And on the other hand, they can also have a better interaction with the fibers. So it can help to increase cholesterol excretion. So in this work, I'm going to focus more on the last two mechanisms regarding the short-chain fatty acids and the biosalts. And we aim to evaluate the coffee melanoidins fractions fermentability and to study how they can influence these biotransformations by the bacteria. So to obtain these rich fractions, we started by doing a dialysis with the coffee brew freeze-dried. Then we obtained the high molecular weight material soluble. And we did several ethanolic precipitations in order to obtain the soluble material in a 75% ethanolic solution, which we call the ETSN, which is the melanoidins rich fractions. And then we characterized these fractions. After that, we did an in vitro fermentation with human fecal microbiota. We had 3 healthy volunteers. We made a pool. And then we mixed with colon nutrient medium for the growth of the bacteria and with the melanoidins fraction. We also had a negative control without the sample. Then we took aliquots at 0 and 48 hours of fermentation, and we analyzed the sugar content, the ammonium production, short-chain fatty acid production, and the secondary biosalts. So looking for the fermentability of this fraction, we can see that before the fermentation, it had 30% of sugars, and it was mainly composed of galactose residues, arabinose, and raminose. And after the fermentation, the 48 hours, the bacteria consumed all these sugars, so the content decreased to more or less 10%, and the profile remained more or less the same. We also looked for the ammonium production, to screen for the proteolytic activity of the bacteria, and we can see that in the first 6 hours, the bacteria was mainly consuming the sugars. And after the 6 hours, it went from the saccharolytic pathway to a proteolytic, producing more ammonium. Regarding the short-chain fatty acids, we can see that they significantly increased after the fermentation of these fibers. And the most prevalent ones were the acetic, propionic, and butyric acid. And as I mentioned, it is important to determine this ratio. And we saw that with the melanoidins rich fractions, the ratio of acetate and propionate was lower, which meant that this fraction produced more propionate, which was the one that's responsible to inhibit the enzymes in cholesterol synthesis. As for the biosalt biotransformations, we saw that in the presence of this extract, the total secondary biosalt production decreased. So, in one hand, even though the dietary fiber can interact better with these secondary biosalts, they are more effective in solubilizing cholesterol, and they can also be toxic for our colon cells. So we don't want a higher amount of these secondary biosalts. So to conclude, we saw that the fermentation of these melanoidins fractions yield a lower ratio of acetate propionate, which have the potential to decrease hepatic lipids. And also, in the presence of these melanoidins fractions, the production of secondary biosalts was lower. So as a main conclusion, the in vitro colonic fermentation of this melanoidins rich fraction has the potential to regulate cholesterol homeostasis. So I just like to thank my co-workers at the Glycofood Chem group in Portugal, and also from the Biociencia in Madrid, to all the funding, and to you all for the attention. Thank you.
Yes, thank you so much. Interesting research. My question directly would be, do you also have some of these fractions in normal coffee and standard coffee types? Or is it necessary that you do the enrichment to get these effects?
For the biosalt sequestration, I haven't showed here, but we have some studies with the coffee beverage and also with the fractions isolated. And we also are trying to evaluate the impact of the processing of coffee, of the roasting and of the grinding, but it's all a preliminary study. But we think that these fractions have a higher effect maybe than the coffee beverage as a whole. So we can either use the coffee as we already take it, or we can enrich in order to have a higher amount of these compounds to potentiate the effect.
Okay, thank you. Are there questions from the audience?
Just a side comment. Thank you for your presentation. But the first chart, I've looked at the caffeine content, but it didn't seem to be proper. I mean, it's the first. It's a side of it because it doesn't really mean much in your presentation. But I think you should check the numbers in your first chart because I don't think they're correct, proper.
Thank you. Thank you. But I don't remember actually, but I know that it can vary a lot depending on the...
Yeah.
It was far from being the normal thing.
Okay, maybe it was a lapse.
But that's not really mattering.
Are there any other questions? We can check up this probably.
I cannot go back now to see it, but...
So,
thank you.
There are no other questions, so thank you again.